Hey, at last we are allowed to start with the laboratory exercises, and we will. It has been some different startup with this because I have done the first steps of the exercise, but we will come back to that. What we will do in this exercise, or both exercises in this course, is to run a meta barcoding study. Meta barcoding is described in chapter 3, so I recommend you to read that very carefully. Both meta barcoding and these metagenomics and these other aspects there. Meta barcoding is a situation where we collect a sample with a mix of a lot of different organisms, or a lot of samples, various samples with different content. Illustrated here. We collect a sample which already is full of various individuals or various organisms. We don't know which species are present. We isolate DNA from this, and in order to identify which species are present, we will run a meta-barcoding study. We use PCR, this DNA amplification technique, to amplify a specific gene, just a small region of the genome, not the entire genome. Then it would be called metagenomics. But meta-barcoding is looking at a specific gene region in the genome of the organisms. We amplify this genome, this gene or this region, and we DNA. We amplify it with the PCR, and then we DNA sequence the amplified material. If we find just one sequence in this result of the DNA sequencing, then we will know, well, the entire sequence is identical. That means it must be just one species in this sample. Of course, if we had chosen a gene or chosen a region with very conserved DNA sequences, a lot of genes are very conserved, so human has the same sequence as another eukaryote, then it of course will be no help for us. We must choose a gene region that shows a high degree of variation, so that all individuals in one species share the same sequence, and all individuals from another species share another DNA sequence. There might be some sequence differences also inside or between the individuals in one species, but we usually say that the intraspecific variation, as described in chapter 3, intraspecific variation, the gene variation inside a species is lower than the interspecific DNA variation. The interspecific genetic variation means the variation between the species. Okay, after the sequencing is finished, we can study our sequences, and we can sort them into groups of various sequences. All the sequences that have this blue sequence belongs to one species. All these brown sequences belongs to another species, and the green, and so on. So depending on how many species that were present in the sample, we will get various number of these pools of or these folders of different sequences. So that is the plan for the entire exercise. This week we will come to this step. We will isolate DNA, we will run PCR, and next week we will run the sequencing part of the study. Right. First, some bullet points connected to this exercise, or to the exercise we are going to do today. We have this this word community DNA, or environmental DNA, or eDNA. Well, it's quite similar words, but and also quite similar meaning. But there is a small differences. Community DNA we often use if we collect a sample which consists of a mixture of DNA from various organisms, various individuals. Then we might call it for community DNA, because it's collected from the same community, from the same in the same sample, and the DNA present in the sample is often more or less the entire genome, or all the DNA from the organisms. For instance, if we take a plankton sample, using various equipment to collect, and then we will have a mixture of a high number of individuals, and also a mixture of a lot of different plankton species. Then we could call this for a community DNA. And environmental DNA is also a mix of DNA from various organisms or various individuals collected in the same sample. We don't know how many individuals, we don't know which species or anything. But environmental DNA might also be just partly degraded DNA, because it comes from cells that has been released from the organisms, and the cells might be partly degraded. The DNA comes out, or it is free DNA. So it is a mixture of free DNA, cells, or partly degraded cells. So that's environmental DNA. What is common between these two? It is a mixture of DNA from several individuals, and might also several species, collected in one sample, and we don't know which species, how many individuals, how many organisms that are present. The metabarcoding we talked about in the last slide, and also in this chapter 3 is full of information about metabarcoding. So please read that. The collection of samples is a point that we should pay attention to, because if you are interested in study the genetic variation or the biological variation in location, you will collect samples from the location, and you will perform a metabarcoding study to identify which species are present, how many, or at least what is the situation of the genetic variation between these species. So you have to collect samples, of course. And then you must ask yourself, is it enough to just collect one sample and then study that? Or should you collect several samples, several locations in the same area? Do you want to collect samples regularly throughout the season or throughout the year in order to have a representative collection of samples, so that it is representative for the for the area. And so that is something that can differ from project to project, but you should absolutely, of course, be aware of that regarding how many samples, how often, and how should I collect them. Another thing in this point is, what do I do with the samples after I have collected them? Is it possible to treat the samples right away, start the DNA isolation, for instance? There's a lot of kits and a lot of methods and equipment available where you can actually start the DNA isolation out in the field where you have collected your sample. The point is that you should make sure that when you start to isolate the DNA, and at the end you will sit with a tube with DNA from this sample, it should contain, of course, the same content, or the DNA from all the individuals that were present in the sample when you collected it. So make sure that the sample will not start to degrade or be in bad quality before you start to isolate DNA. And that can be done in various ways. You can freeze it if you have possibility to that. You can put it in a cooling bag or in a box of ice, or you can add some preservative chemicals. You can add ethanol in some cases. Other cases there are other chemicals that you can treat your samples with in order to make sure that your DNA will not start to degrade. The samples for this study, or this lecture, or this laboratory exercise is just one sample collected from two different environments. One of the environment is compost, that is soil or earth from a compost bin. So that is one sample, and we want to see how many different organisms or different microorganisms, actually how many different bacteria species are present here. The other type of sample was collected from computer keyboards from, well, how many? 4 or 5 offices around at the institute. I took a swab and put on some buffer on the swab, and then I collected sample with the swab from the keyboards from these 4 or 5 different keyboards. That is the 2 samples we have. If we had started earlier with the laboratory, you could have collected your own samples, of course. Okay, next thing is, what should we do with these samples before we start DNA isolation? Could we isolate DNA directly from the sample, or should we start some growth of the microorganisms in order to make sure that we have enough material to get DNA from it, which is possible to sequence? In this case, we have chosen to do some growth, and of course, in this compost sample, it could be possible just to start DNA isolation right away, because there is a lot of bacteria there. And in the keyboard situation, I don't know if it could be possible to isolate DNA, but the best is to start some enrichment procedure. Of course, a dangerous or a risky thing with that will be that some microorganisms will grow happily on a bacteria media plate or in some liquid media, but other bacteria or other organisms are very difficult to grow at the laboratory. So we run the risk that some species will not amplify or not grow in this media. So we might lose some species because of that. In those cases, it could be good to just start sequence right away. Right. So what has been done now is that before this lecture or this exercise, lab exercise, I have done some growth of the microorganisms, both from the compost and from the keyboard samples. Right, the last point here is DNA isolation and barcoding. That means the sequencing and stuff. But we also, the first thing is what you are going to do today. You want to do the DNA isolation. We will go through step by step the protocol, and you will have the protocol. You have downloaded it maybe, and you can you must of course have it at the laboratory, and I will go through it and explain some of the steps. The barcoding, maybe it was a little confusing to say barcode here because we also call it meta barcoding there, but barcoding is also used as a word for tagging the DNA, because now you are several groups or you will work one by one, so you have 8 or 9 groups today, and then you will have 8 or 9 different samples. And if we could have tagged your DNA, the DNA that you will isolate, and when you are sequencing it later, you can identify which sequence was as a result of sequencing of my DNA or the DNA that I put into it, because all DNA will be mixed, and if we tag it or barcode it, we can identify that later. We will talk more about that soon. This is the procedure that you will follow for isolating DNA from your sample. You collect the sample from the laboratory bench and you isolate DNA by using this kit. Power feces DNA kit procedure or power feces DNA kit. It's designed for isolation of DNA from stool or feces, which is a mixture of a lot of both microorganisms, but also some other material that might influence your samples. But it is also very nice for isolating organisms from other samples, as in this case, because that too contains a lot of microorganisms with different properties, both gram-negative and gram-positive, and they might have different, they demand different ways of isolation for getting good results. And this kit is good for all these organisms. You start the isolation by putting your sample into a tube which contain some dry beads. I will come back to that. And in addition, you have some liquid, some solution that makes sure that the cells, the bacteria, will erupt, and that you will have your DNA out. Some bacteria are very difficult to lyse because they have strong cell walls. So these dry beads will do that job, because we add the beads, we add the solution that help the cells to lyse, and we heat them. All these steps will lyse the cells or help for lysing the cells. In addition, we will not use this vortex, but we will use a machine that will shake the tubes vigorously, and this shaking will make sure that the beads that are already in the tube will smash or beat the cells, so it will more easily lyse. So after this step, the cells has lysed. or actually it says in this step, but in this process here where you both beat the cells, you have all these solutions, and you incubate it at the end, then the cells has lysed. After that, we add some more solutions and we incubate it and so on. These steps will make sure that the cells has lysed, and all this other stuff, that some fatty acids and some other molecules will be separated from the liquid where the DNA is present, in order so we have started the process of purifying here. And in this step, we add the solution containing DNA into a spin column, and in this spin column we have a filter, and the DNA will stick to it or bind to this filter. We have the right salt concentration in the solution, and when that is optimal, the DNA will stick to the filter, and all other molecules will not stick to the filter. So you understand what is happening, and some of you have done this before in some different kits and different samples, but the principle is the same. We purify it somewhat in these steps, and then we add the solution with the DNA into the column, and we centrifuge it, and the DNA will stick or bind to the filter. We wash it, that means we send some solution through the filter to remove any molecules that are not DNA there from the filter, so it will be pure. At the end, we add a different solution to the DNA filter situation, and the DNA will lose or will not bind to the filter anymore. So we centrifuge it and we collect the DNA in the collection tube. Then we have isolated and purified the DNA. Right, this is the protocol in details. The protocol that is already in Canvas or put out for you is a more simplified version. Says this exactly the same, but here you see there are some notes or explanation of what is happening at this step in the protocol. So those steps you can read carefully, and we will briefly mention some of them. The first 1 and 2 points has already been done, because I have put the bacteria in tubes for you and added some power bead solution, so that there are some liquid there, and they also start to act on the DNA. And then you will add solution C1. A lot of the solution in these kits, it doesn't say exactly what the content is, because it's secret, I guess. But they have these names, C1 and C2 and C3 and so on. But it is informed about the main ingredients, of course. The C1 solution contains SDS. And SDS is actually a soap, and you know what soap do? It does. It binds to fat and make that the fatty acids will be disrupted. And there's a lot of fatty acids in cell membranes. So this C1 will help breaking down the cell membranes. We heat it to 65 degrees, so this other parts, these polysaccharides and lipids and salts and so on, will also disrupt. And in this pane, put it on a vortex adapter and so on, we will not do, because we have this other instrument that you will see at the laboratory. So we do some centrifugation. We take this supernatant, the thing where after the centrifugation you will have a pellet, and you transfer the supernatant to a clean tube. Expect between 400 and 500 microliter. Make sure that you are not transferring anything of the pellet. Next step, add another solution where it says that you can incubate it for some minutes, and you will do. Then you centrifuge again, and you will have a new pellet and liquid pot, and then you will transfer the supernatant into a new tube. And it is, as it says here, avoid the pellet. So up to 600 microliter, that means that you will not take more than 600 milliliter here. You will use the micropipette and put it to 600, and you take out the volume and make sure that you are not touching the pellet or getting some of this pellet material over to this collection tube. Then you add C3 solution to this collection tube, or to the tube where you transfer the supernatant, and you vortex it briefly just to mix it well and incubate it for 5 minutes in a rather cold environment. There will be a box for you where you can put it for these 5 minutes. After new centrifugation, there will be a new pellet. Avoid the pellet again and transfer up to 750 microliter. Make sure that you will not take more into a new tube. So there's some pipetting steps in this procedure. And here there are some explanation of what is in this pellet. As you see, some inorganic and organic material and so on. We don't want that into our DNA samples. Next step is to add 1200 microliter of C4 solution, vortex, and now it is prepared for adding into this centrifugation column. There's high salt concentration in this C4, so that this impure DNA solution is now high salt content, much salt in this solution, and make the DNA possible to stick or bind to the filter. And then you add 650 microliter of this solution, because there's not room for any more than 650 milliliter in this spin column. Centrifuge it for 1 minute, and then you throw away the flow through, the material that has gone through the filter, and the DNA that was there is now stuck to the filter. And then you continue and 650 microliter into the spin column, centrifuge, and then you throw away what has come through. Put the column back into the tube and add a new 650 microliter, centrifuge again, throw away what has come through, and at a third centrifugation with 650 microliter. Will end up with that you have centrifuged the entire volume through the column, and all DNA is now stuck to the filter. This step, you add 500 microliter of solution C5, which is a washing solution that washes away all molecules that are still present on the filter, and which is not DNA, of course, because DNA has stuck to the filter, so that will not be washed off, but all other molecules and impurities will be washed off in this step. And here you just centrifuge one more time with no adding of any new buffer. That means that is done because we will be completely sure that this C5 solution is away from the filter, because there's some ethanol there, and that is not good to have present on the DNA when we later want to take it out. Right, so after this centrifugation, we carefully transfer the spin column into a new tube. Make sure to avoid any liquid of C5 onto the spin column. and we are almost finished. We add 100 microliter of solution C5. C6. C6 is this elution buffer, which is only a very weak buffer with salt, the weak salty buffer, which will make sure that the DNA will not bind to the filter anymore and be eluted into a collection tube after a centrifugation step. Right, and after this, you have isolated DNA from your sample. All the way, you should make sure that you which kind of sample are you analyzing? Are you analyzing a compost sample with bacteria from compost, or are you studying the keyboard or the K sample? You should note at a piece of paper or wherever, which is because you should know that when you are going to study your sequences later. So when you are finished, you can place the tube in a box of ice. You can put up your initial, so you will find back your tube, and you should also label it with K or C, indicating which kind of sample you have worked with. Right, the last thing you will do is to run the PCR. PCR, of course, you should know in detail what is happening in PCR. We have talked about it in the lecture. This temperature shifting program will make sure that the DNA, even if you have very little DNA in the start, you have amplified your gene or your fragment or your part of the genome as you have chosen. In this case, we will amplify 1,500 base pair fragment, which is covering a gene coding for the 16S rRNA molecules. That is that gene will produce an RNA molecule that will be a part of the ribosome of the bacteria. And there's a lot of sequence differences in this, in a part of this this gene, so that you can compare the sequences, and you will say that, well, this is 16S rRNA gene from this bacteria or from that bacteria. So we amplify it with PCR, and later we will sequence the same. So today you also will run this PCR. You will start the PCR and then you are finished. So later when the PCR is finished, I will pick it up and store it for next week. Important, do not label your tube with a pen. The PCR tubes you must not write on with a pen or an ink or whatever, because it will be in a PCR machine afterwards, which will be almost 100 degrees hot and warm, and that is not good when you have written on it with a pen. So you should just make sure which tube is yours. You take a PCR tube and you will mix these ingredients. You will mix DNA, that is the DNA that you have isolated. If something happens in your isolation procedure, I have also some DNA from these samples that we can use. You take 10 microliter of genomic DNA into a tube. I will come with some information from this on the laboratory, so we will come back to that. But you have 10 microliter of DNA, you add some water, some extra high quality water that you will get, of course, and you will also have a 1 microliter of 16S barcode. A 16S barcode is some kind of DNA tag that will attach to your DNA with a specific sequence. Later on, when we are sequencing it, we will know that, oh, this sequence comes from the CSC sample, that comes from a compost sample. Or when we have sequenced it, we will find another sequence tag, and we will know that, well, this sequence comes from a keyboard sample. So that is what we call with we mean with a 16S barcode. It is a small piece of DNA attached to your DNA. So later on, when it is sequenced, we will be able to sort them into two groups: the compost sequences and the keyboard sequences. We could also have had a different kind of barcode saying that this sequence comes from group A and the students in group A, and this sequence comes from students in group B, and so on. But in this case, we are just dividing into two groups: the compost sample and the keyboard sample. When you have mixed DNA, some water, and these barcodes, you can come to a station where I will add this DNA polymerase enzyme master mix, which is the enzyme that will build the DNA in the PCR process, in the PCR steps. In addition to enzyme, you will have the PCR primers, as you remember what is doing, and also some nucleotides, because when we are building DNA in the PCR, of course, we need nucleotides to produce DNA molecules. So there's enzyme, there's primers, there is nucleotides, and there is a suitable buffer for this. So when this is mixed, you will place your PCR tube in a specific tray in a box, and I will show you where it is. And you will, as I said, don't write anything on your tube, but you should, at the piece of paper beside this box, you will write down where in the tray your sample is located. You will get it then when we are going to sequence this later. So then the day is over, because then I take the samples, put it into the PCR machine, and we will run this temperature profile as this: 25 cycles of PCR with denaturation, high temperature, annealing. The primers will attach to the DNA, and extension. The enzyme will produce new DNA. And after that is finished, you start over again, and it goes 25 rounds of PCR. And when it is finished, it will cool down to 4 degrees, and the samples can be stored in the freezer until next week. And that is the exercise for day 1.